So section 7.5 is covering fermentation and anaerobic respiration. And if you recall from regular cellular respiration, the main thing that produces the ATP is the oxidative phosphorylation, which obviously, since it's oxidative phosphorylation, requires oxygen. So the question that this section tries to answer is what happens when there's no oxygen present? Without oxygen, you have seen that the electron transport chain will no longer operate because, as you know, in the electron transport chain, oxygen is the terminal electron acceptor, which starts that production of ATP. But without the oxygen to accept the electrons, you will not get the rest of the chain and you will not get that ATP. And so the ETC cannot occur. So in that case, glycolysis couples with either anaerobic respiration or something called fermentation to produce ATP. But both of these processes happen without oxygen. They're anaerobic without oxygen. So the first type of respiration that occurs without oxygen is anaerobic respiration, and this is when an alternative electron acceptor is found and the ETC does occur, but maybe with a different molecule such as iron or sulfur. And this is pretty rare for large organisms. Mostly the only organisms that can do this are small prokaryotes that live in an area without oxygen. And so, like I said, they can still undergo the electron transport chain, but some other molecule that's not oxygen is used as the electron acceptor, and in a way, oxidative phosphorylation still occurs and they can produce ATP. The one we're going to focus on, though, more is fermentation, and this is a way that we can harvest chemical energy without oxygen or the electron transport chain. So as you recall, glycolysis is an anaerobic process, and it oxidizes a molecule of glucose to produce 2-pyruvate. And in that process, NAD plus is the oxidizing agent. So we need NAD plus for glycolysis to occur. So one of the problems of anaerobic respiration is that we will run out of NAD plus, because once all of that's been converted to NADH, glycolysis has to stop. So fermentation is a process that allows us to regenerate the NAD plus, which can be reused and glycolysis can continue. And there are two types of fermentation. So one is alcohol fermentation and the other is lactic acid fermentation. But in both of these processes, electrons are going to be transferred from NADH to pyruvate after glycolysis, and that's what's going to help us regenerate our NAD+. So that way we don't run out of it and we can keep going through glycolysis. So alcohol fermentation will be the first one that we talk about, and it takes place mostly in yeast and certain microbes. And essentially what happens in alcohol fermentation, there's two steps. So first, carbon dioxide is released by the pyruvate, and it becomes a 2-carbon compound. You don't need to know this, but that compound is called acetaldehyde. And then that 2-carbon compound is then converted into ethanol, and it's reduced by NADH. So then in the end, NADH releases the NAD+, which, as you know, is what we need for glycolysis, and we can start glycolysis over again. So ethanol is only a 2-carbon molecule, so that's why we end up with our carbon dioxide in the end as well. That gives us our 2 ethanols, so that's 4 carbons, and then 2 carbon dioxides is 6. And remember, we had 2 pyruvates, which were 3, so 6 stays with 6. And we use this process a lot, actually, to make alcohol. So you would see this in, for example, wine making, beer making, etc. We use these microbes, and through their fermentation, that's how we get that alcohol, the ethanol right here. And this slide is straight from the textbook, but it's just another way that you can take a look at this. In bread making, we use yeast that CO2 that gets released in the alcohol fermentation process is what causes your bread to rise. That's why bread has holes in it, because those are the air bubbles from the CO2. This is a picture of what's happening in alcohol fermentation. So remember, from glycolysis, we get our NAD plus is reduced to NADH, and in that example, we also get our pyruvate. So for alcohol fermentation, that pyruvate loses to carbon dioxide. We end up with our acetaldehyde, and then that goes back to ethanol, and we get NAD+. So that way our glycolysis process can start again, and we can get more ATP right here. And the acetaldehyde gets reduced by this NADH that was produced from glycolysis. So that's important as well. The other type of fermentation is known as lactic acid fermentation, and in this case the pyruvate is the final electron acceptor, and that pyruvate gets reduced by NADH, which produces lactate, but there's no release of carbon dioxide. We use lactic acid fermentation to make cheese and yogurt, so the lactic acid fermentation by fungi and bacteria helps us make cheese and yogurt. And also, it's worth noting that our human muscle cells use lactic acid fermentation to generate ATP when oxygen is low. So for example, if you're going for a long sprint, generally the oxygen in your blood level is not sufficient to sustain aerobic cellular respiration, and so you'll switch over to anaerobic respiration. However, something that was recently discovered is people used to believe that this lactic acid buildup is what caused muscle soreness, but they've recently discovered that your blood cells carry the lactic acid away pretty quickly, and so that muscle soreness is probably more likely due to other factors such as strain on your muscles, not actually the lactic acid, which is something new that I learned. So this is a picture of what happens when we have lactic acid fermentation. So once again, glycolysis takes our glucose and we get ATP, pyruvate, and NADH. And in this case, the pyruvate is our electron acceptor, and in accepting those electrons, which as you know come from the NADH, we end up getting lactate, and NAD plus is released. So once again, we have our NAD plus that can begin our glycolysis reaction, and we can continue getting ATP. So just as a little bit of a comparison between aerobic and anaerobic respiration, remember there's three pathways we can use to get ATP. So aerobic cellular respiration is the one we spent all the last few days talking about. Anaerobic respiration is the term they use to refer to those small prokaryotes that can use another molecule instead of oxygen for the electron transport chain. And then fermentation includes lactic acid and alcohol fermentation. So all three of these use glycolysis as a process to oxidize glucose and other fuels to make pyruvate, and that gives all of them a net ATP of 2. So they all, bare minimum, can produce 2 ATP. And in all three, NAD plus is the oxidizing agent that accepts electrons during glycolysis. So NADP is used in glycolysis. However, the oxidation of NADH, so we can replenish our NAD plus, is what's different between the two. So in fermentation, the final electron acceptor is an organic molecule such as pyruvate. This is our alcohol fermentation, this is our lactic acid fermentation, or acetaldehyde, that's our alcohol fermentation. And then in regular cellular respiration or aerobic respiration, when there is oxygen present, you know that the transfer of electrons from NADH is passed through the electron transport chain where oxygen, the aerobic part, can accept them. And the other major difference is, of course, the ATP produced. So regular cellular respiration in the presence of oxygen produces about 32 ATP per glucose, whereas fermentation only produces 2 ATP per glucose, because we know that fermentation can only happen after glycolysis. So the ATP we're getting from those is coming from glycolysis. This is a kind of a picture that shows those comparisons as well. So if we have oxygen from pyruvate, we can move into the mitochondria and begin the citric acid cycle, which will eventually take us to the electron transport chain and oxidative phosphorylation. And when there's no oxygen present, all of these processes remain in the cytosol, in the cytoplasm, and we end up with our alcohol fermentation, our lactic acid fermentation, or maybe some other products. So that's a picture version. So one important way, this is mostly talking about energy so far, which is covering Big Idea 2 on the AP exam, but it's important also to note how cellular respiration connects to Big Idea 1, which is all about evolution. So one interesting thing to think about is our ancestors on Earth, so those prokaryotic cells. These cells, we know, didn't really have many organelles, if any at all, because they didn't have membrane-bound organelles. They could not carry out several processes such as the Krebs cycle, because as you know, those take place in the mitochondria. And we also know that early life on Earth did not have oxygen in the atmosphere, and so we had to get energy from food, but without oxygen. So if you think about that, if you're thinking about the whole process of cellular respiration and how we harness energy from our food, the only one that doesn't require oxygen is glycolysis. And so the thought is that evolutionarily, prokaryotes that evolved glycolysis are the ancestors of all modern life. And one of the pieces of evidence we have for that is that glycolysis is an incredibly widespread process. Almost all living organisms and their cells still use glycolysis, whether they're the smallest bacteria to complex organisms like us. All of them use glycolysis. Some other evidence for the fact that glycolysis was the origin of modern life, besides the fact that it's the most common pathway among all organisms on Earth, and the fact that there was low oxygen in the atmosphere, it's also the fact that glycolysis occurs in the cytoplasm or the cytosol, because as you remember, those prokaryotic cells didn't have the complex organelles like the eukaryotic cells did. So without mitochondria, they wouldn't have been able to undergo the Krebs cycle or the electron transport chain. But because you could undergo all of the processes of glycolysis in the cytosol, you didn't require any organelles, which means prokaryotic cells very well could have undergone glycolysis. And that first oxygen in the atmosphere that we're talking about didn't appear till years after the first fossils of bacteria have been found. So that's an interesting piece about glycolysis.